let us look at what we call light perception. This is basically about how the stimuli, which is basically light, resulting into action potentials or electrical signals being sent to the brain. We are able to see things and say this is a pen, this is a laptop, because of the light that comes from that object and then enters our eyes, and then signals are sent to the brain, and then the brain can interpret what we are seeing. So we want to look at that process. How does it really happen? We already know the cells that are involved in this process, and we call them photosensitive cells, or we also call them photoreceptor cells. Okay, and these are basically the cones and the rods. Therefore, in this presentation, we are going to see how the rods act on the stimulus, which is light, and then convert that into electrical signals. And then we shall, after that, we'll look at how the cones can act on light and also send signals to the brain for interpretation. So we'll basically notice differences here and there, and we should be able to explain those differences. Those differences will lie in terms of clarity, they will lie in terms of sensitivity, they will lie in terms of color perception. Okay, so be very keen on those explanations for those key differences. So photoreceptor cells are just like any other receptors. They act on the stimulus and convert that stimulus into electrical signals or electrical impulses that are interpreted in the brain. Okay, so that means that light in this case is the stimulus, and then the cells that pick that stimulus, which is light in this case, are the ones that we call the photoreceptor cells or photosensitive cells. Okay, so basically what happens, just like what happens in other different receptors, the process is that there should be a change in the environment that can excite the receptors. So that change in the environment that can excite the receptors is what we call the stimulus. The plural of that is stimuli. Okay, so the receptors are able to pick that change through a process that results into generation of what we call generator potentials. Okay, generator potential has to be generated, and that generator potential should have the intensity that is required to reach or to exceed the threshold value for an action potential to come in. So the process is the stimulus, generator potential, and then action potential, okay, which is the electrical signal, or which are the electrical impulses that we're basically talking about, or nerve impulses basically. So the cells that pick up this light and get excited, that is, they get to fire electrical impulses in terms of light. It is the rods and the cones that are found in the retina. Okay, so we have already looked at how they are arranged in the retina, and what are the key differences in terms of structures of these photoreceptor cells. So here we are basically focusing on the functional aspect, not the structural aspect. Though in some of our explanations, you will see that we shall link the functional aspect to the structural aspect. So let us start by looking at the rods. The rods, you can see that structure is rod-shaped, and they have membranes that contain the photopigment or the photosensitive pigment, and that photosensitive pigment is what we call rhodopsin. Okay, so that means that in our discussion, for whether we are talking about cones or rods, we are going to talk about photosensitive pigments. Okay, so what happens to the photosensitive pigments is very important, because it is what generates the generator potential and also the action potential eventually. Okay, so in here, in the outer segment, we have membranes that contain rhodopsin, which is the photosensitive pigment that we're talking about. Okay, so that rhodopsin is actually composed of two components. We have the complex protein component, that is one, and then the second one is retinol, what we can call retinene. Okay, so two things make up rhodopsin: the protein, which is called opsin, okay, or scotopsin, and then retinol. Those are the two things that make up rhodopsin. Remember, retinol is a derivative of vitamin A. That's why we actually need vitamin A for proper vision. Now, retinol exists in two forms. In the cyst form, when it is dark, okay, when we don't have light stimulation, and then it exists as trans when we have light in there. Okay, so what happens if there is no light? Basically, there is no light stimulation. That means no light stimulus. So we are not firing electrical impulses in this state. So the retinol in this state is in the form of cis. What is very important is that you don't want to have action potentials being fired unnecessarily. The action potentials have to be fired when we have the appropriate stimulus, but also the generator potential that comes from that stimulus should be one that reaches the threshold value such that action potentials can be generated. So when we don't have any light stimulation, we are not firing impulses. Okay, the rods are not firing impulses. And what actually helps in that issues to do with the sodium ions. Sodium ions are actively pumped out of the rod's inner segment. Okay, and this causes a decrease in the negative charge inside the rod, around 70 to negative 70 millivolts to about negative 40 millivolts. Okay, and that actually is what keeps helps in keeping the rod in not firing. The other thing that is also very important, that the rod cell releases glutamate, which is a neurotransmitter. This also prevents the bipolar neurons from again firing. So it just keeps the bipolar neuron again in the resting potential, preventing any action potentials from being fired. Okay, so that means that in that state, that means without any slightest stimulation, we are not having impulses being sent, partly because of the sodium ions that are actively being pumped and making the outer segment more. It decreases in the negative charge inside the rod, and then the fact that we have glutamate that is being released. It is a neurotransmitter, and this prevents the bipolar neuron from firing action potentials. Now, what happens when now there is light? What happens is that when we have light, the light will strike the outer segment of the rod. The outer segment of the rod, that's where the light will strike. Because remember, the outer segment is what lies towards the surface of the retina. So the light strikes basically there. Okay, so in that outer segment, that is where you have those membranes that contain the rhodopsin, which is the photosensitive pigment. Now, when light strikes rhodopsin, rhodopsin actually absorbs that light. Okay, so that absorption of that light causes rhodopsin to change. Okay, to change its shape. That change in the shape of rhodopsin is what results into an impulse. Okay, is what results into a generator potential, and if the intensity is sufficient enough, then you have an action potential being fired. That is basically a nerve impulse being sent to the brain via the optic nerve, for interpretation. So that is what happens for the rods. Now, what I did not mention in this slide is that actually the photochemical reaction when rhodopsin absorbs light is that it actually bleaches. Bleaching. That means it is split into its components. Okay, opsin and retinol. Okay, that is what we call bleaching. Okay, so that means you have the rhodopsin being split as a result of absorption of that light. So those